Okay, well, thanks very much to Yaniv and to Tom and the organisers for the invitation to come and speak here today. It's my first time in Israel and it's been fantastic, so yeah, really appreciate the opportunity. Today I'm going to talk about some work that we've been doing trying to understand how the anatomy of the brain constrains its function. And this is something that's been studied quite extensively over the years using lots of different techniques. This is one of my favourite examples. So in this study they looked at two species of worms, C. elegans and P. pacificus. And these two worms have got identical pharyngeal nervous systems, so they've got the same neurons, same types of neurons, but the connectivity between them is different, as you see in the middle, in this projection here. And in this study they showed that differences in the connectivity of just two neurons were sufficient to produce a change in behaviour such that P. pacificus is a predator of C. elegans. So you can see pacificus happily eating elegans for lunch. So this is quite a profound example of how quite minor changes in structure can influence system dynamics and behaviour. And now we can't do the same thing with humans. We can't lock people in a room and see who eats who. But we can do things like map structural and functional connectivity and correlate them. We can use models of dynamics, and we can apply virtual lesions to the models to see what happens if we impact certain brain regions, what happens to activity. Or we can look at clinical case series. We can look at patients with lesions to specific areas and try to map the effects of those lesions on brain activity and behaviour. And all this work has been really important for helping us build this understanding of how specific anatomical properties relate to function, but it's a little bit piecemeal. We sort of study one or a couple of brain regions at once, and we don't really have a unifying framework for understanding how dynamics arise from an underlying structure. Now, in a lot of areas of physics and engineering, you can get insight into structural constraints on system dynamics by considering the eigenmodes of the system. We just heard about them in the last talk. And eigenmodes are obtained by solving this kind of eigenvalue problem. You've probably seen some variant of this. It's used in all sorts of different contexts. But just to remind you, an eigenvector is something that we can measure about a system that will change its scale but not its orientation when subjected to a linear transform. So what we've got here is we've got some vector here, psi, subjected to a transform, and once it's transformed, it's the same vector just scaled by some factor. So this lambda is the eigenvalue, and if this condition holds, then psi is an eigenvector. Now, when we're talking about eigenmodes, or we're talking about fundamental structural properties of a system, so it's probably not correct to think of this as a transform. This is more an operator, and the operator we typically use is a Laplacian operator, which generally describes how smoothly some structural property of the system varies. And I'll come back to different ways of defining that. But for now, I just want to emphasise that the eigenvectors of the Laplacian are orthogonal. So they represent a basis set, as Andrew was saying earlier, that we can then use to reconstruct any spatial pattern that we see in the system. So a classic example is a Fourier basis set, where you can take any arbitrary one-dimensional signal and reconstruct that as a weighted sum of sine and cosine waves of different frequencies. We can do the same for the brain. We can pull out its structural eigenmodes. They give us spatial patterns that vary over different wavelengths. So the first one is always a kind of global mode. Then you have an anterior-posterior, dorsoventral. And as you go higher and higher, you pick up fluctuations on more fine-scale wavelengths. And so then we can take, say, an activation map, such as this one here, Y, and represent that as a weighted sum of our modes with the weights given by the A coefficients here. Now, if that definition or explanation is too technical, you can do what one of the students in my lab did and ask ChatGPT. And it turns out that it gave a really good explanation, so I just thought I'd share it with you. So, explain eigenmodes for a 10-year-old. Certainly. Let's imagine we have a wobbly, jiggly ball of jelly. When you poke it, it wiggles and shakes, right? Now, imagine if you poke the jelly in different spots or with different amounts of force. Each time you poke it differently, the jelly wiggles and shakes in a different way. Eigenmodes are like those different ways the jelly wiggles and shakes when you poke it, right? And this is really fundamentally true. Eigenmodes are used a lot to study vibrations in systems, and the eigenmodes correspond to the preferred patterns of vibrations when you apply energy to the system. And they're determined by the the physical and structural properties of that system. So things like the shape of the jelly, how thick it is, are going to determine the way in which it can vibrate when you apply energy. So if we generalize the context, we can move beyond just vibrations and think about how systems get excited. And really the eigenmodes define preferred patterns of excitation in a system of connected elements. And so this basic concept has been applied to extract eigenmodes of the connectome, as Andrew was just discussing. So the basic idea is we map connectomes with diffusion MRI, we represent that in the form of a graph or an adjacency matrix, and then we compute this property here. It's called the graph Laplacian. It quantifies how smoothly connectivity varies as you move from node to connected node in the network. And so really the eigenmodes of this Laplacian are really correspond to, you can think of them as axes of variation in the connectivity of the network. And several studies have shown that you can use these modes to reconstruct different patterns, the spatial topography of classic resting state networks, different patterns of functional connectivity, various EEG signals, and even patterns of neurodegeneration in different types of dementia. So this work has been really seminal in establishing this as a proof of principle, that this eigenmode framework is quite useful. And importantly, it gives us a direct link to the underlying generative process, because we can start to interpret all of these dynamic patterns we see as representing excitations of the fundamental resonant modes of brain anatomy. Now this work follows what I would very much characterize as a classical view of brain organization, and that's that you can divide the brain into discrete areas that are specialized in some way, and those areas are connected according to some complex array of fibers, and activity is dominated by signals moving through this web of connectivity. Now there is a different way of thinking about things, and that's been developed over the last 50 or so years in the context of neurofield theory. So neurofield theory is a family or class of mathematical models for studying mesoscale and macroscale dynamics, so really spatial scales of above 0.5 millimeters. And the underlying assumption or overarching theme of these models is that we view activity as being dominated by wave-like propagation, travelling waves of excitation. And that's what you see experimentally. If you record the activity over a wide enough field of view at the right resolution, you see that this is voltage-sensitive dye recordings of the dorsal surface of a mouse cortex. You see that you get these travelling waves of excitation moving continuously through the cortex. You're not seeing these little bursts or discrete jumps of activity that would be implied by the model on the left. So neurofield theory really tries to explicitly capture these spatiotemporal patterns of propagation. And in doing so, it doesn't try to divide the cortex into discrete regions. It treats the cortex as a continuous sheet. And depending on the specific form you use, some of the more popular forms make an approximation of connectivity such that each point influences the activity of nearby areas in a distance-dependent way. So activity in one point will influence the activity of nearby areas, and the influence decays roughly exponentially as a function of distance. Now, if you're someone that's spent the last 10 to 15 years worrying about all the intricate connectivity of the brain, this seems like a heresy, right? How could we throw away all that detail and just use this very simple approximation, treat all areas using an isotropic kernel? But there is a lot of evidence to suggest that this simple exponential distance dependence can explain a lot of features of brain organization. So I guess the question is, well, how far can you get with this approximation? Now, one thing that has fallen out of this theory is that if we buy into neurofield theory, we treat the cortex as a continuous sheet, we think wave dynamics are important, we approximate connectivity using this isotropic kernel, then we don't need to derive our eigenmodes from a map of the connectome. We can derive them simply from a model of the geometry of the cortex. So in this case, our operator becomes something called the LaPlace-Beltrami operator, and it describes how smoothly the geometry of the cortex varies in space. So we're treating the cortex as a 2D sheet embedded in 3D space, and therefore we use the LaPlace-Beltrami operator. And this derives directly from the wave equation that's used to summarize dynamics in the model. This is a basic form here. The details are not so important. I'll just say this is how the field propagates in space, and you can see our friend the LaPlace-Beltrami operator pop up here. Now, this equation hasn't been pulled out of thin air. You can start in neurofield theory from first principles, looking at the biophysics of neuronal populations and move up and kind of derive this equation. But this is a fairly generic wave equation that is used to describe wave dynamics in all sorts of different systems. And so the link between geometry and wave-like propagation has been understood for several centuries now, well, 2 centuries. Now, it's also well known that the eigenmodes of the Laplacian in a system dominated by waves correspond to the standing waves of the dynamics. So what are standing waves? Once again, Wikipedia helps here. They arise from the interactions of different travelling waves. So here we've got 2 different travelling waves, a green and a blue, and their pattern of interactions elicits these large-scale amplitude modulations shown in red here that are fixed in space. Right? So that's why they're standing waves. But the amplitude modulations are much larger than the amplitudes of any of the original carrier waves. So this is an example of resonance. It's where we get a larger response from the system than the energy that we put in. And so this is just providing an example of how the eigenmodes of brain anatomy represent the fundamental resonant modes of the dynamics. Now, if you don't believe Wikipedia, here's an example of a real system. Here we've got a pool and the experimenters apply a vibration to a certain part of the wall. The wave travels across to the other side, it hits the opposite wall, then it reflects back, and you can see now the emergence of these standing wave patterns. So they're fixed in space and they just alternate in amplitude at a level that's much higher than the original wave. Another classic example is the Clubny Plates. If you put dust particles on a plate and then you apply a vibration of that plate at a specific resonant frequency, you can elicit these complex patterns. And again, the patterns that you get, they correspond to the eigenmodes of the plate and they're determined by the physical properties of the plate. So things like the stiffness of the material, but also the geometry. So if we had a different shaped plate, we would end up with different patterns. And this is a bit gratuitous, but I just think it's fabulous. You can extend or generalize this concept to 3 dimensions. So here they're using eigenmodes of acoustic waves and they're using it to levitate particles. So I think if I saw this in high school, I would have definitely become a physicist. But I think it's very cool. But you know, this is more than just a passing curiosity. It does have real-world consequences. This is a video of the Tacoma Narrows Bridge, I think around the 1940s in Washington State, USA. The engineers didn't pay enough attention to eigenmodes, and on this particular day, crosswinds hit the bridge in just the right spots and at just the right frequencies to cause these resonant responses, these really large-scale, large-amplitude fluctuations or vibrations, and it ultimately led to the collapse of the bridge. So eigenmodes are important. But we're faced with a choice. If we want to look at them in the context of the brain, what do? We've got 2 different views saying that we should generate our eigenmodes in different ways. We've got the classic connectome-centric view, where we view the cortex as comprising these discrete areas. Dynamics are dominated by node-to-node transmission. We think that connectivity is anisotropic and topologically complex, and we want to capture all of that complexity. We're not really focused on the physical constraints of the brain. We're not really considering spatial or geometric constraints. You can do that secondarily, but it's not baked into the model. And because we're really interested in connectivity, then that means we need to map the connectivity. So we would derive our basis set using a combination of T1 and diffusion MRI data. The other view, which is a geometrically informed view supported by neurofield theory, is that we treat the cortex as a continuous sheet. We're not dividing it up into discrete areas. We think dynamics are dominated by propagating waves of excitation. We make the approximation that connectivity is homogeneous and distance dependent. That's not necessary, but in the simplest, most popular forms of neurofield theory, that's what's done, and so we started there. The model directly accounts for the effects of space and geometry, so it's really prioritising these physical constraints. And a nice practical benefit is because we approximate connectivity, we don't need to map it. We can extract our eigenmodes simply from a model of cortical shape, which we can extract just from a T1 weighted scan. So we wanted to compare how well these different modes were able to reconstruct different patterns of activity. So this was the basic framework. We take different patterns of resting state or task-evoked activity, so spatial map Y here. We represent that as a weighted sum of our eigenmodes. And yeah, we did this for both rest and task. So here is an example of the seven main task activation maps from the Human Connectome project data, different tasks, social, motor, gambling, and so on. And here is the accuracy that this is how well we can reconstruct the maps. So here, the y-axis is the accuracy of the reconstruction, just a spatial correlation. The x-axis is the model order. So as we move along the x-axis, we reconstruct using the first mode, the first two modes, the first three, the first 50, and so on. And so remember, because the modes are ordered by wavelength, as we add more modes, we're better able to capture the finer scale structures in the maps. So if we start with 10 modes, we're already doing okay. We've got correlations of about 0.5. It's kind of capturing very coarse scale features, but misses a lot of the details. At about 100 modes, we're doing quite well. We're at about 0.8, and we're capturing really the essential features of the maps. And you can go out to 200 modes, and you'll capture pretty much most of the relevant detail, but the increment in accuracy is not huge. And we see the same with resting state data. We can reproduce resting state functional connectivity data, and about 100 modes is enough to pick up the key features of the matrices. So I'll just emphasize that this is purely using the geometric modes. So all we've got is T1 weighted data, and we can reconstruct these different functional patterns. But of course, the question is, well, how does that compare to the connectome modes? So here is a comparison with respect to the resting state data. The black line is the geometric modes. The purple and green are different variants of the connectome modes, and you can see across all model orders, the geometric modes are outperforming the connectome. And then we looked at 47 different task activation maps you can generate from the Human Connectome project. So each row is a different task contrast. We subtract the difference in accuracies between geometry and connectome. So red means geometry is doing better. These are different variants of the connectome modes, and you can see that generally geometry is doing better across the board. And of course, connectomes are sensitive to the way in which you parcellate the data, to the way in which you threshold the data, and so we confirmed that this did not have an impact. We see the same results regardless of what you do. And just out of interest, it wasn't a main point of comparison, but we find that the geometric modes are more generalizable than statistical modes derived from the functional data itself. So we did a PCA within a given task, and that will give you a linearly optimal basis set for that task, but then we ask, well, how well can that be used to reconstruct other tasks? And it turns out the geometric modes do much better on that kind of outer task generalization than the task-specific PCA. So, hopefully that's a bit of evidence to kind of get you thinking, oh, well, maybe there is a fair amount of information about function just in the geometry or the shape of the cortex. The nice thing about this eigenmode framework is that it allows you to look into the spatial frequency content of the activation data. And just to illustrate why that might be important, consider what we normally do when we're mapping task-related activity. We'll run some statistical analysis. We end up with, say, some unthresholded map of t-statistics here representing areas of increased activation. Then we can project that onto a 1D line. So this is our statistic value, and this is moving through space, and we see some pattern. Then typically we'll apply a threshold, and we only then consider the bits that are above or below the threshold, and we see these clusters here, and we say, these are the areas that are involved in my task. But of course, if we applied a different threshold, we'd end up with a slightly different conclusion. But hopefully it's evident from this sort of projection that we're really only capturing the tips of the icebergs. We're missing the structure of this more global pattern. And so the eigenmode framework allows us to capture that full structure and characterize it in detail in a way that's similar to a power spectral decomposition of a time series, the temporal data that you might do with a Fourier decomposition. We're doing this now in the spatial domain, just by looking at the weighting coefficients of the different spatial eigenmodes. And so when we do that for the different task activation maps in the HCP data, we find that most of the power is concentrated in the long wavelength modes. So this cutoff here represents eigenmodes with wavelengths of about 60 mil or greater. Right? So suggesting that these different tasks are really eliciting or exciting these brain-wide modes that vary over fairly coarse spatial wavelengths. And just to confirm this wasn't something specific about the HCP data, we also looked at over 10,000 different activation maps in the Neurofield repository. So these have been recorded doing just about anything you would want anyone to do in an fMRI scan, a sensory task, motor task, cognitive task, language, emotion, the whole thing. And we see the same basic pattern. This is an average across the 10K, but if you look at individual maps, it's all pretty consistent. So it suggests that regardless of what you're getting people to do, brain activity, as recorded with fMRI, tends to excite these long wavelength brain-wide modes that really challenges this classical view or approach of really trying to focus on these isolated clusters of regional activation. And we were able to confirm that this wasn't just due to an effect of the way in which the data are processed or smoothed. It can't be explained by that. And we also show that if you selectively remove the long wavelength modes from the reconstruction model, the accuracy declines much faster than if you remove the short wavelength modes. So again, just showing that it's really those long wavelength modes that are driving the spatial patterns of activity. And we see this across all the tasks. So, remember that I said the reason why we think geometry might be informative of function comes from Neurofield theory, where we're assuming that wave dynamics drive a lot of activity. We wanted to explore this a little bit further, so we just used a simple wave model. So again, this was the wave equation I showed earlier. Again, with Neurofield theory, you can start from a detailed biophysical model and move up to derive this equation, but we ignored all that detail. We just said we just want a generic wave process, and we want to see how much of fMRI data we can reconstruct. And we compared that to a popular kind of state-of-the-art neural mass model. So these are used quite extensively in the literature. Again, the basic idea is you divide the brain into discrete areas. You have a model, a biophysical model of the local population dynamics, and then you link those populations up according to an empirically mapped connectome, and they start to influence each other, and then you can simulate patterns of functional connectivity. So we wanted to compare how well these two models could account for different aspects of brain activity. And I just want to point out there's a very big difference in model complexity. So the wave model only takes the T1 geometry as input and has two parameters, one that's fixed, one that's fitted to the data. The mass model, the mass model requires T1 and diffusion MRI map of connectivity and has 19 parameters, 15 that are fixed and 4 that are fitted to the data. So when we actually see how well they can explain just static edge-level functional connectivity, we find that they're performing pretty much the same level, the wave model at the top, mass model over here. When we look at regional averaged functional connectivity, we find that the wave model is doing slightly better. And when we look at functional connectivity dynamics, so how the network evolves through time, we find the wave model is doing a fair bit better. So it's better able to capture the way the dynamics of the network through time. And in response to a reviewer comment, we also looked at lagged interactions between brain regions. We found the wave model did much better than the mass model. So again, suggesting, again, because of this difference in model complexity, the wave model offers a much more parsimonious account of these different resting state dynamics. Now we also looked at evoked dynamics. We just did a simple thing where we applied a stimulation in the wave model to V1, visual cortex. And it's interesting, you trigger this wave of activity moving from the back to the front of the brain, and what you see is you naturally see this segregation into dorsal and visual processing streams, right? We also get a hierarchy of response timescales, whereas you move the cortical hierarchy, you see longer and more prolonged responses in different areas, which correlates quite nicely with T1-T2 ratio, which has been validated as a reasonable non-invasive proxy for cortical hierarchical rank. Now the reason I say this is because these properties, things like dorsal segregation of dorsal ventral streams, hierarchy of timescale responses, these have been studied quite extensively using both theoretical and experimental data and have been thought to depend on complex patterns of layer-specific connectivity. But these data are suggesting that that's not necessary for these complex patterns to emerge. Waves and geometry are sufficient to produce these. Now that's not to say the connectivity is not important, not doing anything. It just means that this is a kind of minimum set of sufficient conditions to get these complex patterns of activity to emerge. So I'll just wrap up by emphasising everything I've talked about to date has been on the cortex. So that raises the question, what happens in subcortex? So I wanted to look at how similar the geometric modes were to the dominant functional modes in these different areas. So here I've got the first 3 non-uniform geometric modes of the thalamus. And here are the first 3 functional modes generated using a dimension reduction of the fMRI data itself. You can see they look very similar. So if you do a scatter plot, it looks very impressive. And we see the same pattern if we look at the striatum and hippocampus, all areas with very different geometries. And this correspondence extends out to the first 20 or so modes. So suggesting there's a really tight coupling between geometry and function in these areas. And I just want to point out that these functional modes, these gradients, are obtained using a really elaborate data processing pipeline. You start with the time series in all of your subcortical voxels, you correlate those with voxels outside the subcortical ROI, then you subject that matrix to a dimension reduction, and then incorporate that and generate a similarity matrix between every pair of subcortical regions. Then you subject that similarity matrix to a manifold learning algorithm, and then you obtain your gradients. We're showing here that we can get pretty much the same information just by taking the shape of the area and decomposing that, which takes just a couple of minutes. So again, it just points this really close link between geometry and function in these areas. So hopefully I've convinced you that eigenmodes offer a nice formal framework for linking brain structure and dynamics. It aligns with approaches that have been used for a very long time in physics and engineering and gives us a nice direct generative interpretation of the activity we see as resulting from excitations of fundamental modes of anatomy. The data I've presented suggests that modes derived from geometry represent a more parsimonious anatomical basis set for brain function than those derived from the connectome, and suggests that there's a lot to be gained by prioritizing physical properties of the brain, such as space and geometry. This kind of framework suggests that different types of stimuli evoke brain-wide responses of long wavelength modes, and so it really tells us that maybe we're throwing the baby out with the bathwater in a way by just focusing on these really focal areas of activation. And the modelling suggests that travelling waves, homogeneous connectivity and geometry are sufficient to explain quite a diverse range of apparently complex dynamical patterns. And so in a way it represents a bit of a baseline that we can then use to start testing hypotheses about what more complex aspects of connectivity contribute to brain function. So I just close by emphasising that all of this work was done by 2 brilliant postdocs, James Pang and Kevin Aquino. So please direct all praise and criticism, mainly criticism, to them. And also a great collaborators, Ben Fulcher, Peter Robinson, who developed a lot of the theory over the past 3 decades, Michael Breakspear and Mariana Alder-Hinkle. And thank you for your attention. We have time for questions?
Thank you for your talk. I study EEG, and there is a EEG corresponding part of this global theory, which was initiated by, I believe, Paul Nunez in 1974. And in his model, the conduction speed is defined by the thickness of the white matter, and the thicker the faster, right? And that determines, well, not only that, but that is part of the, some of the factors that determines the traveling speed. And from that, you can predict the mode, right? In your fMRI-based model, what determines that traveling speed? And what is, in other words, what's the medium of this wave phenomenon?
So if I understand the question correctly, so the question is, well, what's the speed of the wave?
Yeah.
Yeah. So I mean, when you're just doing the eigenmode expansion, you don't need that because the modes just come from geometry. In the wave model at the end, that was the parameter that was fitted to the data. So we fitted the wave speed. No, hold on. Sorry, one of the parameters is, I forget now which one. One of the parameters is wave speed, and I can't remember if we fixed it. There was one that was wave speed and there was one that was length scale. I'd have to check the paper, sorry.
Hi, up there. So really exciting work. I'm sort of wondering, so you're assuming that all your interactions are local. And as we know, the brain is very not local. So did you think of also trying to add an additional term to the wave equation to look at less local, maybe a square term or something like that, to look at less local interactions, and how does that affect all of the dynamics?
Yeah, I mean, I think it depends. We need to be a bit careful about what we mean by local. So there's an exponential distance dependence, but the length scale of that dependence still allows for quite long-range connectivity. So I think, in a way, actually, here's something I prepared earlier. So, you know, if you look at, say, a micrograph, you can see these penetrating fibers coming in from the white matter, but you can see that there's a lot of horizontal connections, right? And so, you know, let's kind of expand that out. Typically what we do when we do our discrete connectomes is we plonk some regions on top and we completely miss all of that horizontal connectivity, and we just look at the bits that are kind of penetrating the white matter. But as it says, as I've got the stats up there in V1, 80% of the synapses are horizontal, right? So by Neurofield theory really prioritizes capturing those, and it just makes a simple approximation of the bits that move into the white matter. So you're still able to capture long-range interactions. Now, there are forms of Neurofield theory where you can incorporate more heterogeneous patterns of connectivity, and this is something that we're interested in looking into, like how much do you gain by adding that in? But in the first case, we just wanted to start with the simplest approximation.
Question? I have two questions. So I guess you contrast this geometric view on the propagation of activity in cortex with connectionist view, where the wave propagation or eigenmodes are based on the connections. But I mean, there is actually a link between them, which is wiring economy. So I mean, it's known that areas in cortex and in general in the brain brain regions are located, if they're connected, they're located close to each other. So I expect that even if you look at the mesoscopic connectivity, it will appear local, right, in space. So I don't really see much. I mean, there are subtleties, right, in how local regions are connected, but like on a very gross level, it would appear to me that they're similar, meaning that regions which are connected, they are located close to each other in space.
Yeah, so here's also something I prepared earlier. So, you know, if you plot connectivity strength as a function of distance, this is diffusion MRI data, you see there's this big exponential trend, right? And this has been shown in all sorts of animals, just about any scale you look at, there's this big effect, right? And I think a lot of it is due to saving wire. It's a metabolically efficient way of connecting areas. Not everything is explained by it though. As you can see here, there are these connections in blue and red that kind of deviate from the trend. I guess a lot of connectomics, including my own work over the past 10 to 15 years, has focused on those red and blue bits and, you know, tried to understand, well, why are they there? They seem to be important. What does it mean? But at the same time, you've got to realize that that's a really small fraction of all of the connections in the brain. So perhaps it's not so surprising that we can explain a lot of function just by focusing on that big dominant trend.
Yeah, I guess the second question is a bit more critical. So if you have a phenomenon which is those traveling waves on the brain surface, which are independent or kind of unrelated to connectivity, does it mean that connectivity is irrelevant to brain function, or it means the phenomenon is irrelevant to brain function?
I wouldn't say that the waves are not dependent on the connectivity. So they are. They're just supported by a specific form of connectivity, right, which we can approximate using a simple exponential kernel. Now, you can have wave dynamics with more complex patterns of connectivity, but, you know, I guess the point of this work was to show that we can use this simple approximation and get very far. So now the next question is, well, what happens if you incorporate this more complex connectivity?
All right, Alex, thank you very much.